Ever taken a look at, like, a glass of water and wondered what's actually going on there?
It's crazy to think.
I mean, we can't really see anything, right?
Mm.
But there's a whole world happening at a microscopic level.
Oh, absolutely. It's a whole universe we rarely even think about.
Right. Like we hear bacteria and instantly think, ew, germs, stay away.
Yeah.
But seriously, there's so much more to these little guys. Some of them can even, like, swim around.
It's true. They've got their own little world down there, and that ability to move, to actually navigate their environment, that's what we call motility.
So how do scientists even figure that out? I mean, we're talking about something you can barely see under a microscope. Are we, like, attaching tiny little GoPros to them or something?
Haha, I wish. Though the technology for that would be incredible. No, we rely on a couple of really cool techniques to figure out if bacteria have what it takes to be called motile. One of the, I guess you could say classic methods is the wet mount.
Wet mount. Sounds a little messy.
Not at all. It's actually way simpler than it sounds. Picture this. You're in the lab, got your microscope all set up. You take a teeny tiny drop of liquid, could be water, a bacterial culture, whatever you're looking at, and you put it on a glass slide.
Okay, so far so good.
Then you just cover it with a thin cover slip. Think of it like putting a little protective shield over your tiny drop. Boom, wet mount.
And that's it. Then we just stick it under the microscope and watch these little guys do their thing.
Well, almost. You see, there's a difference between true motility, like actual swimming, and what we call Brownian motion.
Brownian motion.
Yeah, it's basically the way things move randomly in a fluid because they're constantly getting bombarded by these tiny little water molecules.
So they're not really swimming. It's more like they're getting jostled around.
Exactly. It's like being in a mosh pit, but on a microscopic level.
Yeah.
The water molecules are constantly bumping into the bacteria, making them jiggle and bounce around.
Oh, okay. So it's like a microscopic rave.
Kinda. So the real trick is being able to tell the difference between that random jiggling, the Brownian motion, and true motility, where the bacteria are actually propelling themselves.
So we're looking for the bacteria that are doing their own thing, not just going with the flow of the microscopic mosh pit.
Exactly. You want to see purpose. Like, are they just randomly vibrating, or are they making a beeline for something, maybe a food source or something? That kind of movement, that's a good indicator of true motility.
Okay, so let's say I'm watching this little bacterium laser focused on, like, a sugar crystal. How is it even moving itself? It's what, like 1,000,000 times smaller than a grain of sand?
It's pretty amazing when you think about it. Bacteria have these tiny little structures called flagella. They're kind of like little whips or propellers.
What, do they actually have propellers?
Well, not exactly propellers, but you get the idea. These flagellas spin around and propel the bacteria through their environment.
So what makes these, like, microscopic motors run? Do they run on sugar?
That's a great question. They actually run on something called a proton gradient. It's a bit complicated, but basically it's a difference in electrical charge across the bacterial cell membrane. It's like a tiny little battery that powers those flagellar motors.
Wow, a built-in battery.
Yeah.
These guys are amazing. But if it's just a motor, wouldn't they just go in one direction forever?
That's where it gets really cool. Bacteria can actually change the direction their flagella rotate, which lets them tumble around and change direction.
So they have, like, a bacterial reverse gear?
Pretty much. They've evolved this incredibly sophisticated system for sensing their surroundings and navigating through them. But, you know, sometimes wet mounts aren't enough. Like, what if the bacteria are really slow movers?
Oh, right. We might not even be able to tell they're moving.
Exactly. That's where our second motility test comes in, the semi-solid medium test.
Semi-solid medium. Okay, now you're just making stuff up.
Nope, it's a real thing. Imagine your typical Petri dish with that kind of gelatinous stuff in it. That's agar. It's used to grow bacteria. A semi-solid medium is similar, but with less agar, so it's kind of wiggly.
Wiggly, like Jell-O.
Yeah, kind of like Jell-O. It's firm enough to hold its shape, but loose enough for the bacteria to move around in if they have flagella.
So we're basically setting up a bacterial obstacle course.
In a way, yeah. And to introduce our bacteria to this Jell-O-like medium, we use what's called a stab line. We take a sterile needle, dip it in the bacteria, and stab it straight down into the medium.
A stab line. Sounds kind of violent.
It's not as bad as it sounds. We're just making a starting line for our bacteria. And then we let them grow for a bit and see what happens.
And if they can move, they'll, escape from the stab line?
Exactly. Motile bacteria will spread out from that initial stab, creating a kind of fuzzy halo around it.
And if they can't move, they just stay put.
Yep, they'll pretty much be stuck there, so you'll just see growth along the stab line.
So we've got these two pretty cool ways to spy on our bacteria buddies and see if they're, out there tearing up the dance floor or just kind of stuck in place.
Yeah. It's like the wet mount is catching them live in action, and the semi-solid medium is more like checking out the after-party photos.
Right. So if both tests tell us if bacteria can move, why use both? Why not just, pick one?
Well, think of it this way. Each test has its strengths. Sometimes one method just gives a clearer picture depending on what kind of bacteria we're dealing with.
Okay, I'm curious. Give me an example. When would you absolutely want to use one over the other?
Let's say you're looking at a bacterium that's super speedy, zooms around under the microscope. It'd be really hard to tell what it's doing in a wet mount.
Yeah, like trying to track a fly in a hurricane.
Exactly.
Yeah.
And in that case, the semi-solid medium would be way more helpful. We give those little speed demons some time to move around, and then boom, we can easily see how far they've spread.
So it's like, instead of trying to clock their speed, we're measuring their endurance.
Right. Now, on the flip side, imagine a bacterium that's, not exactly winning any races. Watching them in a wet mount would be like watching grass grow.
Ugh, yeah, I got better things to do with my time.
Exactly. But with the semi-solid medium test, we can just let them hang out and see if they've moved at all after a while. It's a more relaxed approach.
I love it. We're giving these little guys their moment to shine, no matter their pace.
Exactly. And the best part is we're learning so much about this hidden world that's literally right under our noses.
It's mind-blowing. Who knew there was so much going on in a seemingly still drop of water?
It really makes you appreciate the sheer diversity of life, even at the microscopic level.
Absolutely. So next time you see a glass of water, just remember, there might be a whole lot more happening in there than meets the eye. Tiny bacteria, with their incredible motors and navigational skills, could be swimming around, searching for food, maybe even doing the conga line. Who knows what other secrets these microscopic marvels hold? Guess that's something to ponder the next time you're gazing into your water glass, huh? Until next time, keep exploring the unseen worlds all around you.